Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Assalamu alaikum, dear students. Yesterday we have discussed about the various types of ovules. We have seen we have 6 types of ovules. Today we will take up megasporogenesis and development of female gametophyte or embryo sac. This megasporogenesis, it is the process of formation of megaspores from megaspore mother cells. And in the process of formation of megaspores from the megaspore mother cells is called megasporogenesis. It usually takes place inside the nucellus of developing ovule in angiosperms. And in this megasporogenesis, it takes place inside the nucellus, the parenchymatous tissue surrounding the embryo sac, that is called as the nucellus. This megasporogenesis, it takes place inside the nucellus of developing ovule in angiosperms. How this takes place? It takes place by the differentiation of a single hypodermal cell, generally towards the micropylar end of the ovule. And it takes place due to the differentiation of a single hypodermal cell towards the micropylar end of the ovule. And we can say megasporogenesis takes place at the micropylar end of the ovule with the differentiation of a single hypodermal cell. This single hypodermal cell either this directly acts as the megaspore mother cell, or this hypodermal cell, before acting as the megaspore mother cell, it divides periclinally into an outer primary parietal cell and an inner primary sporogenous cell. And we can say here the hypodermal cell, it directly acts as the megaspore mother cell, or before acting as the megaspore mother cell, it undergoes a single periclinal division to produce an outer primary parietal cell and an inner primary sporogenous cell. This inner primary sporogenous cell then acts as the megaspore mother cell. This then acts as the megaspore mother cell. And we can say the process of formation of megaspores from megaspore mother cells is called the megasporogenesis. This generally occurs inside the nucellus of the developing ovule in angiosperms. It begins with the differentiation of a single hypodermal cell which is present towards the micropylar end of the ovule. This single hypodermal cell, it acts as the archosporium. This archosporial cell, that is the hypodermal cell, either this directly acts as the megaspore mother cell, as we have seen in case of tenucleate ovules, or before acting as the megaspore mother cell, it divides periclinally into an outer primary parietal cell and an inner primary sporogenous cell. This inner primary sporogenous cell then acts as the megaspore mother cell. This megaspore mother cell then undergoes meiosis. It undergoes meiosis to produce a linear tetrad of megaspores. To produce a linear tetrad of megaspores. And in this megaspore mother cell, it then divides meiotically, it undergoes meiosis to produce a linear tetrad of megaspores. Occasionally, T-shaped or inverted T-shaped, this megaspore tetrad is produced. And generally this megaspore tetrad is linear. Occasionally, in some cases, we see that T-shaped or inverted T-shaped megaspore tetrad is also produced. But in case of male, we have seen that this megaspore tetrad may be of different shapes. It may be linear, it may be tetrahedral, it may be isobilateral, it may be decosate, it may be T-shaped. But here, in case of female megaspore tetrad, it is generally linear. Occasionally, T or inverted T-shaped tetrad is produced. Now, what happens later? This from this megaspore tetrad, generally the upper three megaspores, that is micropylar three megaspores, they degenerate. Generally the upper three degenerate and the lower one, that is causal one, only causal one, that is the lower one, remains functional. And it is the lower one only which produces the female gametophyte, while as in case of male we have seen all the microspores, they produce the male gametophyte, that is pollen gland. Here we see that only the lower one, generally only the lower one, that is causal one, it remains functional, that is elongates, develops further and develops into the female gametophyte, and the upper three, that is micropylar three, they generally degenerate. Now the functional megaspore, that is the causal megaspore, it divides thrice, and it divides three times mitotically to produce 8 megaspores. These to produce 8 cells. How, we will see. First, this functional megaspore, it divides into 2, and it produces 2 this megaspore, it divides and produces 2 nuclei. One nucleus moves towards the micropylar end and another moves towards the causal end. Actually, first 2 nuclei are produced, then a big center vacuole is produced in the center. This displaces these nuclei towards the 2 ends, that is one micropylar end and another towards the causal end. Then second division takes place. This micropylar one, it divides into 2, and causal one, it also divides into 2. Then another division takes place. Micropylar produces 4 and causal also produces 4 nuclei. Then what happens? And if we get 8 nuclei, one nucleus is divided into 8 nuclei by 3 divisions. What happens to the 4 micropylar nuclei and the 4 causal nuclei? From the 4 micropylar nuclei, 3 organize to produce egg apparatus. 3 organize to produce egg apparatus, 1 egg cell and 2 synergids. These 3 micropylar 3, they produce egg apparatus, this egg apparatus, and out of the 4 which are at the causal end, 3 organize to produce 3 antipodals. Antipodes, as we see. 4th from the micropylar end, it is displaced towards the center, and 4th from the causal one, it is also displaced towards the center. With the result, we have in the center 2 nuclei which organize to produce the central cell. These 2 nuclei which are in the center, they are called as polar nuclei. Polar nuclei, as you have seen here, polar nuclei, or this we also call as secondary nucleus. Because if these 2 nuclei remain haploid, we call them simply as polar nuclei, and if they fuse, they form a diploid structure. This diploid nucleus we call as secondary nucleus, and this cell we call as central cell. Thus we can say that a typical female gametophyte, this we call as the female gametophyte, it has 3 structures, 3 parts: egg apparatus, central cell and antipodals. Egg apparatus is comprised of 3 cells. Central cell, it is made up of 1 cell but with 2 nuclei, which we call as polar nuclei, and third is antipodals, and antipodals are also 3. These egg apparatus is towards the micropylar end, antipodal is towards the causal end. Now one more thing, the synergids of the egg apparatus, they produce some cellular hair-like structures called filiform apparatus. These filiform apparatus play an important role in guiding the pollen tube towards the micropyle and or towards the egg apparatus. And it is these filiform apparatus which guide, which attract the pollen tube towards the egg apparatus, and when pollen tube enters into one of the synergids, when it enters into synergid, it bursts, and the pollen tube secretes two male gametes, and from these two male gametes one fuses with the egg and produces the zygote, and another fuses with the central cell, polar cell, that produces the triploid structure that is called the primary endosperm nucleus. Now we have seen that this typical embryo sac, it is 7-celled. Typical angiosperm embryo sac, it is 7-celled and 8 nucleate. It is 7-celled and 8 nucleate, and all the cells of the embryo sac, they are haploid except the central cell, which is diploid. Central cell is diploid. All other cells of the female gametophyte, they are haploid. Now this type of embryo sac, which is 7-celled and 8 nucleate, this is called as monosporic embryo sac. It is also called as polygonum type of embryo sac, because it was first worked out in polygonum and is the most common type of embryo sac found in angiosperms, and is found in about 70% of the angiosperms. That means the embryo sac which is produced from the single megaspore of a megaspore tetrad, that is called monosporic embryo sac. If only one megaspore takes part in the formation of in the development of the female gametophyte or embryo sac, it is called monosporic embryo sac. And if 2 or all the 4 megaspores of a tetrad, they take part in the development of the female gametophyte, then that embryo sac is called a bisporic or tetrasporic embryo sac respectively. And we have seen now development of first megasporogenesis and development of the female gametophyte, and also structure of the embryo sac. That is, a typical embryo sac or female gametophyte has 3 important parts, that is egg apparatus, central cell, and antipodals. I have already discussed these 3 parts. This is all about for today's lecture. Thank you. Assalamu alaikum wa rahmatullahi wa barakatuh.